talk a little bit about molecular biology. In molecular biology, two nucleotides of opposite complementary DNA or RNA strands that are connected by hydrogen bonds are called a base pair. Ribonucleic acid is one of the three major forms of the micromolecules.
A genetic disorder is a disease caused by abnormalities in an individual's genetic material. There are 4 types of genetic disorders.
This one in 1,250 people. Here's how people are affected.
Brett is copying pages out of a textbook and then sending them out of the nucleus.
Perfect.
Then he arrives at the terminator, which tells it to stop.
A karyotype for a normal person, someone with no genetic disorders, has 46 chromosomes, 23 given by each parent. The sex chromosomes are an X or a Y, 2 X's equal a female, and an X and Y have a male. A normal karyotype has exactly 46 chromosomes. Any more or any less results in a genetic disorder. This is an example of a normal karyotype. The 2 X's equal a female.
3. When we look at this baby's karyotype, we see that it has an extra 21st chromosome. This is a non-disjunction called trisomy 21, or Down syndrome. Down syndrome is an autosomal non-disjunction. The karyotype can help discover genetic disorders even in infant or fetal stages of life.
This is the karyotype of the baby with Down syndrome, with the 3 21st chromosomes.
Karyotypes can be used to discover traits of a person. For example, the gender of a person can be found by examining X or Y chromosomes. Females have 2 X chromosomes and a male have an X and a Y chromosome. Some males have 2 Y chromosomes, which is super male. Kids with this condition may have learning problems. This occurs randomly and does not cause physical appearance differences.
Another hour of yesterday, all I wanted was to walk.
Hey, my name is Morgan Walker, and I'm going to tell you about DNA base pairing along with briefly describing DNA to RNA and DNA replication. In simplest form, you are composed of DNA. Every cell in your body contains DNA, which is found and held in the nucleus. Each strand of DNA contains triplets, which are 3 letters. There are 4 different choices of letters that make up each triplet. You can have an A, C, T, or G. A must always correspond with T, and C must always correspond with G.
There is a common phrase that assists us in remembering the pairing. A and T is where is that, and I'll see G. By remembering this common phrase, you can often recall the DNA base pairing quickly.
Now I'm going to briefly relate DNA to DNA base pairing to DNA to RNA base pairing. When the DNA sends messages out of the cells to the proteins, aka central dogma, the letters that correspond slightly change. Instead of A corresponding with T, as in DNA to DNA base pairing, A now corresponds with T. In other words, there is no T in DNA to RNA base pairing. How do we get more DNA? The double helix inside of your cells is DNA. In DNA replication, the 2 strands which intertwine to make your DNA unzip into 2 new strands which intertwine to make new DNA.
Do you know the difference between homozygous and heterozygous? Homozygous is when the 2 alleles from the trait are the same. Heterozygous is when the 2 alleles from the trait differ from each other. An example of homozygous are those 2 plants. An example of heterozygous are those 2 plants.